Hello students, now we are going to start the next chapter of the biology of the 11th class, that is the cell cycle and cell division. Okay, why we are going to discuss this chapter now? Because as you know, every organism is made up of cells, and cells are the basic structural and functional unit of the life. So these are the cells only that will decide the structure and functions of an organism. But as you know, organisms are divided into two categories, prokaryotes and the eukaryotes. So there is a mode of reproduction that will help to sustain the life on the earth, because that reproduction will help in to sustain the species of specific type on the earth. But question arises, how these species are formed? Yes, if we will discuss about the prokaryotes are the unicellular organisms in which main mode of reproduction is only the cell division. Yes, cell division is the method by which they will make the new cells, and these are the new cells are their only their body. But if we are discussing about the multicellular or the eukaryotes are made up of millions or billions of the cells, because these cells are arranged together to form the tissues, and tissues will make the organs will make the organ system, and different types of organ system of an organism will make a specific structure of an organism. Okay, so how the life will begin in the eukaryotes, as you studied in the 10th class also in the reproduction chapter, that gametes are produced by the organisms. Yes, that is the sperm is produced by the male and egg is produced by the female. These will fertilize together to form a single cell zygote. Yes, this is the single cell zygote, that is the diploid structure, and this diploid structure will then pass through a division that is known as the mitosis, and with the help of this mitosis, that is known as the equational division, because mitosis will maintain the same chromosome number in the daughter cells with respect to their parent cells. So this diploid chromosomes that is found in the zygote, with the help of mitosis, will pass into the various cells, and these cells will then make the different tissues, organs and organ system. That means this is the cell division that will convey the information that is present in the one cell, and that has to be passed into the next cells, tissues, and that will specify the what type of phenotype or structures will be formed in an organisms. So these are the cell division that will correct the information pass from one generation to other generation also, and how this information will be conveyed into other into next generation. That means with the help of the chromosomes it will pass into the gametes, and then gametes will fertilize together to form the zygote. So we will start from here. Yes, that is the Rudolf Virchow, the scientist who first time stated that the cells arise from the pre-existing cells. This thing we have studied in the 9th class also. After that, Fleming, the scientist, was there that given the term mitosis, and the Farmer and Murray give the term meiosis. Mitosis, equational division, and meiosis is the reductional division. We will discuss about it in the detail in next classes. So, is this chapter? Yes, that is known as the cell cycle and cell division. My question arises, what is cell cycle? Cell cycle term was given by the Howard and Paley in 1953. But what cell cycle is? Cell cycle is a process by which a cell will divide to form two daughter cells. Yes, but two daughter cells, how? By multiplying or by duplicating its genome and all the cytoplasmic content. As you know, a cell contains two main things, that is the brain of the cell, that is known as the nucleus, and other parts of the cell that are present in the cytoplasm, that is known as the cell organelles. So when a cell will divide to form the new cells, these components should be divided properly in an equal way. So two steps are required. First, duplication of the genome that is present in the cells, and second, the duplication of the cell organelles which is present in the cytoplasm. So generation time is the time period between the two successive cell division. That means the time period when the cell will start, when cell divide and when that cell which is already divided will again starts to divide, that time period between the two cell division is known as the generation period, and it will vary from organism to organism, because we can say complexity of the organism will vary. It will depend upon the diversity, a huge amount of diversity is present on the earth, and according to that complexity is there, and according to that complexity the cell generation time is also varying. For example, E. coli bacteria is there that will take the 20 minutes to multiply, while as if you take an example of the human cell, it will take the 24 hours to multiply itself. That means, as you know, E. coli is the prokaryotic organism which is a single cell, so it is taking that less time period, that is the 20 minutes, as compared human beings are the complex organisms which are taking the more time period, that means it is taking the 24 hours to multiply itself. So cell cycle is consists of mainly two phases. One is the interphase and second is the M phase. Yes, interphase is the long undividing phase. That means interphase will include all that sequential events which is required for division, but division is not takes place in this. It means it is a long undividing preparatory phase. Yes, that is known as the interphase, where synthesis and growth of the cell will takes place. Second is M phase, where the exactly nuclear division will takes place. After the nuclear division, next phase will be the cytokinesis, where division of the cytoplasm will takes place. So I have drawn a diagram here where I have shown the interphase. This blue line will indicate the interphase that will start from the G1 till the G2. Yes, it includes G1, S phase and G2 phase. After that, this is the M phase that I have written here by the green marker. That includes the prophase, metaphase, anaphase, telophase. This is the M phase. After that, the cytokinesis. So we will discuss about these. First is the G1, that is known as the gap 1 phase. Gap 1 phase mainly include the preparation for the DNA replication. Yes, what are the material required for the duplication of the DNA? As you know, DNA requires the nucleotides, phosphates, sugar, ATP. All these materials will be collected for the DNA replication in this G1 phase only. So I have mentioned also here all these things, and mitochondria plus chloroplast will synthesize in the plant cell. If we will discuss about the second, that is known as the S phase or the synthetic phase. S phase include the replication of the DNA. That means the preparation that already going on in the G1 phase, where material will be synthesized for the replication, but process was still in wait. So that replication process will occur in the S phase. That is why it is known as the synthetic phase, where new chromosomes will be synthesized. That means duplication of the DNA will take place, along with the histones and non-histones proteins will also synthesize. So nucleosome is a structure that is formed here. Nucleosome, yes, where negatively charged DNA is wrapped by the histone protein. That structure is known as the nucleosome. Third phase is known as the G2 phase. Yes, G2 phase, where synthesis of RNA, proteins, centrioles, cell organelles and Golgi body. Cell organelles include the Golgi body and mitochondria. Yes, synthesis of these organelles will take place in G2. So many times there is question comes in the NEET also, that protein synthesis will takes place in which of the phase? Yes, in S phase only histone and non-histone proteins will be synthesized, but in G2 phase translation process will going on. The proteins which is required for the maintain the structure of an organism will be synthesized in G2 phase. Next phase is the M phase, that is known as the metaphase, which include two phases, karyokinesis and the cytokinesis. Karyokinesis means the division of the nucleus and cytokinesis means division of the cytoplasm. One more phase that is left here is the G0 phase. Yes, this G0 phase, which is also known as the quiescent phase. When any cell will not divide, that means when cell do not divide, but it is metabolically active but does not proliferate, it will remains in the G0 phase. Then who will control that cell will divide or not divide? Definitely, there are the checkpoints in the cell. There are two types of checkpoint. First checkpoint that is lies between the G1 and the S phase, and second checkpoint between G2 and the M phase. So I can drawn here also. If any of the checkpoint will we can say if any of the checkpoints will not perform its function, that maybe leads to the cancer. Because cancer is a disease when a cell will divide indefinitely in a uncontrolled way, and this control way is only the checkpoint that will control the cell division. Thank you, beta.